Today, we're diving into one of the most exciting frontiers in medicine. We're talking about the powerful combo of artificial intelligence and proteomics. We're going to see how teaching computers to understand the tiny building blocks of our bodies is about to completely change how we diagnose diseases. Let's get right into it.
Just think about this for a second. What if an AI could peer into the incredibly complex world of your proteins and tell you that a disease is on its way years before you ever feel a single symptom? And look, this isn't science fiction anymore. It's the very real promise of the tech we're exploring today.
So here's what we're going to cover. First, we'll talk about why biomedicine is practically drowning in data. Then we'll unpack the AI tools that are making sense of it all. We'll see exactly how AI is supercharging proteomics, how that's helping us find critical disease markers, and finally, we'll get into the really big questions about the human element and the ethics of it all.
You know, for the longest time, biomedicine was kind of seen as a soft science, but wow, has that changed? Today, it's all about data. We're talking about a virtual avalanche of information, producing gigabytes and gigabytes of data from just a single experiment. And a massive chunk of that data comes from something called proteomics. That's the study of the proteome, the entire set of proteins buzzing around in our cells at any given time. And this is so important because proteins are the little machines doing pretty much all the work. They give our cells structure, they control our genes, they're where the action is.
Okay, but here's the catch. The proteome is just insanely complex. We're talking millions of data points, a sea of information that's just impossible for a human brain to process alone. And that right there, that's the kind of massive, messy puzzle that modern AI was practically born to solve.
So when we say AI, what are we actually talking about? Let's unpack that toolkit, because these aren't just buzzwords. They're actually different layers of computational power, and it's not all the same thing.
Okay, this diagram breaks it down really nicely. Think of artificial intelligence as the big umbrella. Underneath that, you've got machine learning, or ML. That's where algorithms learn from data without you having to spell everything out for them. And then, a super powerful piece of ML is deep learning, or DL, which uses these complex things called neural networks to find really subtle patterns.
Now, there's a really important trade-off you got to understand here. Your classic statistical models, they're easy for us to get. We can see how they work, but they just can't handle a ton of complexity. Deep learning models, though, they are incredibly powerful, but they often act like a black box. We know they work, but we don't always know why they work.
Okay, let's use an analogy to really nail this down. Imagine you want to teach a computer to spot a certain flower. With classic machine learning, you'd have to tell it, okay, look for this petal length and this color and this sepal width. But with deep learning, you just show it thousands of pictures of flowers, and it figures out for itself what the important features are. It literally teaches itself. Pretty cool, right?
All right, so we've got this amazing tool that can literally teach itself. But how does this actually play out in a real lab? Where does the rubber meet the road in the world of proteomics?
So, here's a super simplified look at what happens. You start with a sample, right? Then you basically chop up the proteins into smaller bits, called peptides. Those peptides get separated out, broken up even more, and then they're zapped through this incredible machine, a mass spectrometer, which figures out exactly what they are and how much of each is there.
And check this out. It's not like AI is just helping out in one little spot. No way. As you can see here, machine learning has been involved since the mid-2000s. And since around 2017, deep learning has been woven into pretty much every single step of this process, from start to finish.
Okay, so here's the real game changer. The moment everything really changed was when these deep learning models got good enough to look at the basic sequence of a peptide and predict exactly what its signal should look like when it gets zapped by that machine. This was a massive leap forward, and it made the whole analysis way more accurate and reliable.
Okay, so that's all the cool tech. But so what? Why does any of this matter? Well, it matters because this is how we're finding life-saving clues, or biomarkers, that are hidden deep inside our bodies. And the way they do this now is with something called the rectangular strategy. Instead of just looking at a few samples really closely, they now look at hundreds or even thousands of patient samples and measure a ton of proteins in every single one. It's all about creating these huge, rich data sets, which is exactly what machine learning models love to eat for breakfast.
So, you build a model. How do you know if it's any good? Well, you test the heck out of it. This chart here is a standard tool for that, called an ROC curve. Basically, it shows you how good the model is at telling sick people from healthy people. All you really need to know is you're looking for a score called the area under the curve, or AUC.
Now, to put this in perspective, a perfect score is 1. A total coin flip, a random guess, is 0.5. This model, It scored a 0.96. I mean, that is ridiculously accurate. It shows just how powerful this approach can be.
And listen, this isn't just happening in theory in a lab somewhere. We're talking about real, promising results for some of the toughest diseases out there. Researchers are building new diagnostic models for things like alcoholic liver disease, Alzheimer's, and Parkinson's, conditions where early diagnosis is everything.
Which brings us to our last section, and honestly maybe the most important one. Because with all this incredible power comes huge, huge responsibility. We have to think about the human element.
And it really boils down to two huge challenges. First, we need those black box AI models to be explainable. A doctor isn't going to trust a result if they have no idea how the AI got there. And second, we have an absolute duty to protect patient privacy. This data is incredibly personal, and we need strict controls on it.
So you've got this natural tension, right? On one side, scientists want to share data to speed up discoveries. That's the whole idea behind the FAIR principles. But on the other side, you've got regulations like GDPR, because this data is so personal, it could literally be used to identify you. That's a big deal.
But what if technology could offer a solution to its own problem? There's a really promising idea called federated learning. Basically, instead of bringing all the sensitive data to one central place, you send the AI model on a road trip. It learns at each local hospital or lab and only brings back the lessons, not the personal data. It's a pretty clever way to get the best of both worlds.
And so that leaves us with one last massive question to think about. We're talking about an unprecedented ability to read the source code of our own bodies. So as we move forward, the question we all have to ask ourselves is, as we unlock this biological code, who gets to read it? Who holds the key?